This conference will now be recorded.
Hello students. In new NEET 2024 crash course, now we are in 8th chapter, Cell, the Unit of Life. And in this chapter, we are learning about prokaryotic and eukaryotic cells, then plant cell and animal cell, and the cell organelles, like cell structure, cell function, then the endomembrane system, then endoplasmic reticulum, Golgi bodies, lysosome, vacuoles, mitochondria, ribosomes, microbodies, cytoskeleton, cilia, flagella, centrioles, so all this we are learning. So in total, we are learning about cell and cell organelles. And this chapter is very important for NEET exam. It's having more weightage. In the previous class, we have learned about cell wall and the function of cell wall we have learned. Then about the types of cell, prokaryotic cell, eukaryotic cell, then about cell wall and function of cell wall, and plasma membrane and function of plasma membrane, everything I learned. So here now we are starting with protoplasm. Protoplasm. So what are all points we need to learn about protoplasm? Actually, protoplasm means that's a complex part, right? Why we are saying it's a complex part? And protoplasm is a fluid part, right? It's a fluid part of the cell. So the complex fluid part of the cell, that is called as protoplasm. And we know protoplasm is a fluid and it's a viscous fluid. Viscous fluid, and that is colorless. And this, we can say, this is the living part, or this part can conduct or that can perform all the vital activities, right? Vital activities are performed in this part. And protoplasm, that we can divide or we can differentiate into two regions. Protoplasm we can differentiate into two regions. Which are all the two regions? That is nucleoplasm. Nucleoplasm. What is nucleoplasm? Protoplasm of the nucleus. Protoplasm which is present inside the nucleus. Okay. And next is cytoplasm. Cytoplasm means extranuclear protoplasm. Apart from inside the nucleus, you can see a part of this protoplasm, right? So that is called as nucleoplasm. Apart from that, what and all protoplasm area or the protoplasmic content which you can see inside the cell, that is called as cytoplasm. Okay. Extra nuclear protoplasm, that is called as cytoplasm. Then the cytoplasm, that is covering major portion of the cell. This covers majority of the cell. Majority of the cell, that is occupied by cytoplasm, and that cytoplasm, that is limited or that is separated from the outside environment by plasma membrane. Okay, cytoplasm is limited within the plasma membrane. And inside the nucleus, how it is limited? By nuclear membrane. So inside cell, you can see a nucleus. So inside nucleus, you can see the genetic material, and that genetic material is immersed in what? In the protoplasm, and that is covered, that is limited by a nuclear membrane, right? So inside the nucleus, you can see protoplasm, that is called as nucleoplasm. Then the cytoplasm, that is here outside the nucleus, and inside the plasma membrane you can see the protoplasm, that is called as what? That is called as cytoplasm. So protoplasm means nucleoplasm plus cytoplasm. Okay, and cytoplasm covers major part of the cell, and that is limited inside the plasma membrane. Then cell organelles like mitochondria, endoplasmic reticulum, endoplasmic reticulum, endoplasmic reticulum, then Golgi apparatus. All these are embedded in cytoplasm. So here in the cytoplasm, what and all, you can see endoplasmic reticulum will be there, mitochondria will be there, Golgi apparatus will be there. So all these are present in the cytoplasm. Okay. Now we have learned about what? See, outer covering like cell wall and cell membrane we learned. Then the protoplasm, that also over. Now we need to learn about what? Cell organelles, right? So in cell organelles, what and all we can learn? Cell organelles, their structure and function. Okay, structure and function of cell organelles we need to learn. So here, cell organelles, they are of two types. You can see cell organelles are of two types. What are the two types of cell organelles? Membrane bound organelles and non-membranous cell organelles. Okay, membrane bound and non-membranous cell organelles. Second one is non-membranous cell organelles. Okay, so membrane bound organelles means that is a well organized cellular structure. Okay, and that will be having a unique function also. So endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, lysosome, all these are membrane bound cell organelles. What and all? Endoplasmic reticulum, right? Then Golgi apparatus, then mitochondria, chloroplast, lysosome, all these are membrane bound cell organelles. Okay, then the endomembrane system that consists of nuclear envelope, nuclear envelope, then endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles. All these are suspended in cytoplasm. So endomembrane system means the nuclear envelope, endoplasmic reticulum, Golgi complex, vacuole, lysosome, all these are suspended in the cytoplasm. Okay. So these are membrane-bound cell organelles. Then what about non-membranous cell organelles? The non-membranous cell organelles. Let's see what and all they. See, non-membranous cell organelles, they are also suspended in the cytoplasm and they are associated with the membranous organelles. Okay. And these structures, they are attached to membrane-bound organelles. Okay. And they are also present in the cytoplasm. They are suspended in the cytoplasm. Then which are all the non-membrane bound organelles like ribosome? Examples are ribosome, centrosome, microbodies, then microtubules and microfilaments. Microfilaments. Okay, so non-membranous cell organelles, like they are suspended, they are also present in the cytoplasm and they don't have a membrane-bound structure, and these are attached to the membrane-bound organelles. And examples we need to say like centrosome, microbodies, microtubules, microfilaments, then ribosomes. Okay, ribosome is an example for non-membranous cell organelle. Okay, then now we need to discuss all the cell organelles one by one. So let's discuss first one, mitochondria. See, while learning about mitochondria, you should have a clear picture. You have to learn the structure with diagram. If you are learning, it will be best. Even for NEET exam, you won't get confused. For board exam, you have to learn the structure. But for NEET exam, the best thing is with picture when you are learning, it will be there. It will be always there in your mind. You will not get confused. Okay. And this mitochondria, when we are saying about mitochondria, the first thing which we need to say that who first observed this one? Okay, mitochondria that were first observed by whom? Kolliker. Kolliker, and that was in 1850. Okay, and Altman, R. Altman, he named mitochondria as the bioblast. Named mitochondria as bioblast. Okay, and who gave this present name, the mitochondria? That name was coined by name mitochondria. That's coined by, or that was coined by C. Benda. Okay, then when we are moving to the structure of mitochondria, mitochondria are cylindrical structures. Cylindrical structures, and these cylindrical bodies, they have an average diameter. What about the diameter? 0.2 to 1 micron diameter it will be having, and length. See, that much small bodies they are. Okay. And the length of that body is 3 to 10 micro. That is the length. Okay. Then in mitochondria, you can see that is showing variable shapes. So they are showing different shapes it may show. That's why we are saying that they are pleomorphic. Okay, they are showing pleomorphism. That means it is showing different shapes, and mitochondria will be absent in prokaryotic cell. Where mitochondria is absent, that remain absent in prokaryotic cells. In prokaryotic cells, mitochondria is absent, and in mature human RBC also it's absent. Mature human RBC, red blood corpuscles, in that also mitochondria is absent. This is very important. MCQ. Its mitochondria is absent in prokaryotic cell as well as mature human RBC. Okay. Then mitochondria, we said that they are oval and rod-shaped organelles. Okay, they are the organelles which are having oval and rod-shaped structure, and this is formed by, while drawing the diagram, have you observed we are drawing a double membrane, right? It is having double membrane. So it's having a double membrane structure, and inner membrane is folded into projections. So in the double membrane, the inner membrane that is folded, and that forms a structure called cristae. Okay, the inner membrane foldings, it is forming projections which are called cristae, and this is containing so many tiny particles. Cristae is having tiny particles called oxysomes or F1 particles. Cristae, they possess tiny particles called oxysomes or F1 particles. Okay. Then mitochondria, that is having DNA. So mitochondrial DNA is small. Mitochondrial DNA, that is small DNA, and that is very simple and double stranded, that may be circular or linear. Okay, and the size of the mitochondrial genome, that is very large in plants compared to animals. Mitochondrial DNA is large in plants compared to animals. Okay. Then we used to say that mitochondria, these are semi-autonomous cell organelles. Why we are saying they are semi-autonomous? Semi-autonomous cell organelles because they have DNA and they have ribosomes also. So they are able to synthesize protein. Semi-autonomous. They can do, semi means half of the function they can do, right? So semi-autonomous, they can do itself. So the cell organelles, the semi-autonomous cell organelles we are calling, for which one? Mitochondria. Why we are saying? Because it is having the DNA, the genetic material, and that is having ribosomes also. So they can synthesize protein. Okay. Then next is about semi-autonomous. It is having DNA and ribosome. Okay. Then Krebs cycle, that is taking place in the matrix of mitochondria. Okay, Krebs cycle. in the matrix of mitochondria, Krebs cycle is taking place. See, this is important question. Large. The DNA of mitochondria that is large in plants compared to animals. Then they are semi-autonomous. This is another question. And Krebs cycle that is taking place in the matrix of mitochondria. And the circular DNA, we said DNA is there in the mitochondria. So that circular DNA and ribosomes are also present in mitochondria. So that 70S ribosome, these are present in matrix. So Krebs cycle is taking place in the matrix of mitochondria. Okay. Then what about the mammalian mitochondria? We need to say some points about mammalian mitochondria. So in mammalian mitochondria, 55S ribosome are found, and inner membrane of that cristae that is bearing F1 particles or oxysomes, and that will be having spherical head. So while drawing the structure, we are doing that inside cristae. We are making some particle-like structure on cristae, right? So that is having spherical head, which is subtended by a stalk or the base, which is called as F0. Okay, so F1 subunit of oxysome, that is integral protein embedded in the membrane lipid. And this F1 F0 combination that functions as ATP synthetase, which is catalyzing ATP synthesis. Okay, this is the F1 F0 combination. Okay, F1 particles, or we are calling oxysomes, which are having spherical head and a stalk-like structure. So that stalk-like structure, that is F0, that head-like portion, that is F1. F1 particle or the oxysomes that is having a spherical head, which is subtended by the stalk and a base. Okay. So this F1 F0 combination that is functioning as ATP synthetase. ATP synthetase. What is ATP synthetase? That is the enzyme which is catalyzing the ATP synthesis. Okay, then now let's see what are all the functions of mitochondria. See, these many points you must learn so that you can cover 95% of questions from this mitochondria part. Okay, now let's move to the function. That portion also we need to cover. Functions of mitochondria. So functions, when we are saying about the functions of mitochondria, see, we know that main site for cellular respiration, right? Cellular respiration is taking place in the mitochondria, right? That is the main site for cellular respiration, and ATP synthesis is taking place. So this is a site of oxidative phosphorylation. Okay, so cellular respiration or oxidative phosphorylation is taking place in mitochondria, and that's why it is called as ATP mills of the cell. It's called as ATP mills of cell. Okay, then that is regulating this mitochondria, that is regulating calcium ion concentration inside the cell. Calcium ion concentration inside cell, that is regulated by mitochondria, and that is providing intermediate for the synthesis of chlorophyll, cytochrome, steroid, all these things. Okay, that is providing intermediate for the synthesis of steroid, chlorophyll, and cytochrome. Okay, so these are the main functions of what? These are the main functions of mitochondria, a cell organelle. Okay, and second one, the next cell organelle which we are going to learn, that is about plastid. So here the term plastid, who gave this term plastid? The term was given by E. Haeckel. So E. Haeckel gave the term plastid. Okay, and these plastids, they are the largest cell organelles. Largest cell organelles. Plastids are the largest cell organelles and they have double membrane bound structures, right? So these are double membrane bound structures. Plastids are having double membrane bound structures, and mainly they are used for trapping radiation and storage purpose. This is for trapping radiation and then for storage purpose. Okay, for trapping radiation and for storage purpose, these plastids are working. And these plastids, where and all, see, like these important points you need to notice. Plastids are absent where these are absent in fungi, then in some bacteria, then algae, then in multicellular animals. Okay, and these plastids are classified in two different types like leucoplast, chromoplast, then chloroplast. Okay, so plastids, they are categorized into leucoplast. The first one, leucoplast, then chromoplast, and chloroplast. Okay, and this leucoplast is colorless. Okay, and they leucoplast, that is of three types, which are they: aleuroplast, eleoplast, and ameloplast. Aleuroplast, then eleoplast, and amyloplast. Okay, so this aleuroplast or proteinoplast, that you can uh, see in maize. Aleuroplast you can see in maize, and here aleuroplast or proteinoplast, they can store protein. Okay, so they can store protein and that you can see in maize. Then eleoplast or oleosomes, they can store lipid or fat. Okay, that you can see in endosperm of castor seed. Endosperm cells of castor seed. And amyloplast which is storing starch, that you can see in potato tuber. Okay, amyloplast can store starch, which you can see in potato tuber, and aleuroplast, which is a proteinoplast, and that can store protein. An example is maize. In maize, you can see this. And eleoplast or oleosomes, which have the storing capacity of lipid and fat, and that we can see in the castor seed endosperm cells. Then amyloplast, that can store starch, and you can see that in potato tuber. Okay, then leucoplast over. Next comes the chromoplast. So chromoplast, these are also colorless plastids. Colorless plastid, and these are responsible for the process other than photosynthesis. Other than photosynthesis. Okay, so all the process it will do other than photosynthesis, and different forms are there like rhodoplast, phaeoplast, chromatophores. Rhodoplast. See, the rhodoplast that is found in red algae or rhodophyceae, rhodoplast in rhodophyceae or red algae, and that will be having chlorophyll A, chlorophyll D, then R-phycoerythrin, R-phycocyanin. So all these will be present in rhodoplast. Then what about phaeoplast? Phaeoplast that you can see in brown algae. This is in red algae, phaeoplast in brown algae, and that is having chlorophyll A, chlorophyll C, and fucoxanthin. Chlorophyll A, C, and fucoxanthin. Okay, then last type, that is chromatophores. Chromatophores that you can see in cyanobacteria or blue-green algae. Cyanobacteria or blue-green algae and in bacteria. Okay, so that is about chromoplast, and next is about chloroplast. Chloroplast, it's familiar. Chloroplast is very familiar one, and chloroplast, these are green plastids. Both are this plastids, leucoplast and chromoplast, both are colorless, and here chloroplast, that is green plastid. Right, which one? Chloroplast. And here you know the structure of chloroplast, it is having double membrane, right, and both the membranes are very smooth. The inner membrane is less permeable compared to the outer membrane, and that is rich in protein, like especially we can say the carrier proteins, right, and the space in between two membranes, that is the inner membrane and outer membrane, that is called as periplastidial space. Periplastidial space, and inside these two membrane you can see the matrix which is present in the center, and here thylakoids are closed, like it's like a flat closed bags of membrane which is containing the pigments, okay, which is containing the chlorophyll pigment. Okay, so here double membrane is there, and in between these two membrane the space which is called as periplastidial space, and inside the membrane you can see the matrix, and inside matrix the flat bag like structures are there, that is called as thylakoids, which are covered with membrane, and inside that we can see the pigments, and many membranous tubules they are found in stroma which are called as stroma lamellae. Okay, so many such kind of membranous tubules which are found in the stroma are called stroma lamellae, or we can say Fret channels, and these are interconnecting the thylakoids of different grana. Okay, so this which structure you need to learn chloroplast, then it will be very easy for explanation. Then chloroplast, they are semi-autonomous organelles, right? Because that is containing four components which are very important for the autonomous function, right? Which and all they DNA and DNA polymerase, RNA polymerase, and the protein synthesizing system. Okay, so that's why we are saying it is semi-autonomous. Okay, and what are all the functions of this plastids? Functions of plastids. See, the photosynthesis, which is a very important process, right? The light reaction, dark reaction, all these are included, right? So that is occurring within the chloroplast. Photosynthesis. In that, all the important steps like light and dark reaction, everything is occurring within the chloroplast. Right? Then, in granum, what is taking place? In granum, that is a site of light reaction. Okay. And the matrix, that is the site of dark reaction. Granum is a site of light reaction because in that pigments are present. Inside the granum, pigments are present. So that will absorb the light, right? So light reaction is taking place in the pigment where the pigment is present. That means in the granum. And dark reaction is taking place in the stroma. And in photorespiration process also, this chloroplast is taking part. And this chloroplast, that is the site of glycolic acid synthesis. Glycolic acid synthesis. Okay, then in some cases, this like these plastids, they will metamorphose into chromoplast. For example, in fruits of tomato, you can see. Okay, so these are the main functions of plastids. Okay, next we are moving to the another organelle which is called as endoplasmic reticulum. Endoplasmic reticulum, or ER, we can say ER, endoplasmic reticulum. See, all over the cell you can see a network or a membrane bound, a branch network like structure all over the cell. So that is connecting all the places of the cell membrane and the nuclear membrane also, right? Network like structure, which is called as endoplasmic inside the plasm, cytoplasm, and all you can see that is a reticulate like or network like structure. Okay, so it is a branched membranous network which is spread all over the cell, which is connecting at the place to the cell membrane and the nuclear membrane also. Okay, and this endoplasmic reticulum that was discovered by Potter, Potter and colleagues, Potter et al. Will say. Okay, and in 1945. And there are two types of endoplasmic reticulum, rough endoplasmic reticulum RER, and next type is smooth endoplasmic reticulum or SER. Smooth endoplasmic reticulum, that means without ribosome. Rough endoplasmic reticulum means with ribosome on the surface. Okay, and this endoplasmic reticulum that consists of intercommunicating system or the channels which is making up the membranous sacs. Okay, so each endoplasmic reticulum that is having, or this endoplasmic reticulum system that will be having intercommunicating system of channels, and that is made up of these membranous sacs. Okay, like cisternae, vesicles and tubules. Vesicles, then tubules. Okay, so endoplasmic reticulum is made up of cisternae, vesicles and tubules, and this endoplasmic reticulum that helps in protein synthesis, that helps in protein synthesis. Why we are saying that it is helping in protein synthesis? Because indirectly it is providing space for ribosome, rough endoplasmic reticulum that is having the rough appearance due to the presence of ribosome on the surface. So indirectly it is helping for protein synthesis. So the protein synthesized that will enter into the endoplasmic reticulum and then to Golgi complex, or directly it will be secreted out of the cell. Okay, then functions of endoplasmic reticulum, that also we need to learn. See, endoplasmic reticulum, that is directly or indirectly it is involved in lipid synthesis, protein synthesis, glycogen synthesis, lipid, protein or glycogen synthesis. Okay, and this is involved in the formation of glycosomes and glycosomes and peroxisomes. Involved in the formation of peroxisomes and glycosomes. And this endoplasmic reticulum that helps in membrane biosynthesis, cell secretion, then the transport of ions and transport of molecules, transport of ions, then molecules. For all these, it is helping. So these are the main functions. Then one more important function which we need to say, synthesis of cholesterol, steroid, detoxification of toxins, and it is giving mechanical strength to the cell. So all these are the simple functions or the other supporting function which is given by the endoplasmic reticulum. Next, we need to learn about. So these are the main points which you need to learn about the endoplasmic reticulum. So next, the fourth one which we are learning, that is Golgi body. Okay, so Golgi body, this was discovered by Camillo Golgi. Who discovered? Camillo Golgi in 1898, and he was an Italian scientist, and while learning about the nerve cell, this discovery happened. Okay, so Golgi complex of plants that is known as dictyosomes. Of plants, that is called as dictyosomes. Okay, and dictyosome, this term was coined by Peroncito. Okay, the term dictyosome. That was coined by Peron. And plant cells. See, plant cells will be having several Golgi bodies and they are collectively called as Golgi bodies or Golgi complex. Plant cells have several Golgi bodies and these are together called as Golgi complex in plants. Okay, and there are three main components of this Golgi body, that means cisternae, then tubule and vesicle. Three main parts are there. Which are they? Cisternae, tubule, then vesicles. Okay, so the cisternae means it's a flat and sac-like structure. Then tubules, tube-like structure, which are the seen in cluster, and the vesicles, they are the large vacuole filled with glandular content. Okay. Then we need to learn. So these cisternae, tubules, vesicles, these are the main things which you need to learn. Then what is Golgi complex and who coined this? Or who discovered this, who coined this. So all these things. And apart from that, functions of Golgi bodies you need to learn. Functions. So what are all the functions? Mainly we need to say about two functions like Golgi bodies which are performing so many functions, and that include the absorption of toxic compounds. Toxic compounds. Absorption of toxic compounds, then synthesis of polysaccharide, then acrosome formation, glycolipid synthesis, fluid regulation, then lysosome formation. All these are the functions. Okay, then for protein transfer, it is helping secretion of glycoprotein, then fluid regulation, then cell wall formation, egg covering, absorption of lipids. All these are the some other functions which we need to mention, or which is done with the help of Golgi bodies. Mainly we need to say what and all: absorption of toxic compounds, synthesis of polysaccharide, then formation of acrosome, synthesis of glycolipid, then fluid regulation. All these are the then formation of a lysosome. That is also an important function of Golgi body. Okay, then the supporting function where and all it is doing, like fluid regulation, cell wall formation, absorption of lipids, egg covering, protein transfer, secretion of glycoprotein, and all these Golgi bodies are doing supportive function also. Okay. Next cell organelle, that is lysosomes. Okay. So this lysosomes, this was discovered by D. Dew. Sorry, name D. Dew. Discovered by D. Dew, and how that got discovered in 1949, biochemical experiments, he was doing all those things in the rat liver. So related to that, discovery of this lysosome happened. Okay, and he also introduced this term lysosome. Okay, and they said lysosome, they are called as suicidal bags of the cell. Suicidal bags. Suicidal bags of the cell because they have hydrolytic enzymes. Why they are calling? Due to the presence of hydrolytic enzymes. They have the hydrolytic enzymes, so it is called as suicidal bags of the cell. And this lysosomes, they are the organelles which are bound in single membrane. They have single membrane. It is bound in a single membrane, and that is found in animal cells. Found in animal cells except mature mammalian RBC. Except, this is important question, where it is found? It is found in all the animal cells except mature mammalian RBC. Which one? Lysosome. Okay. And these lysosomes are absent in plants. In plants, it is absent, and in animals, almost all the animal cells, it is present except mature mammalian RBC. Okay. And this lysosome, we said it is having hydrolytic enzymes. So these lysosome, they'll release this hydrolytic enzyme in damaging the aged cells, to kill the aged cells or to digest those cells by the process called autolysis. Releases the digestive or hydrolytic enzymes, and that digests the old cells, and that process is called as autolysis or autolysis, killing of the cells. Okay. So these lysosomes, they are always called as autophagosomes, called as autophagosomes or scavengers of cells. Okay. And these lysosomes, they are also helping in the cell division. Okay, here they're helping in the cell division. That also we need to mention. So Dedew, he discovered this, and they're called as, lysosomes are called as suicidal bags because they have the hydrolytic enzyme and they will digest the old cells. Okay, or the cells which are to be removed. So that process is called as autolysis, and they have lysosome that is having single membrane, and it is found in all the animal cells except mature mammalian RBC. And this lysosomes are absent in plants. They are called as autophagosome or scavenger of cells, and these lysosomes, they are helping in cell division also. Okay, in the process of cell division, it is helping. Clear? So today we have learned about the protoplasm, then about the membrane bound cell organelles like mitochondria, plastid, then endoplasmic reticulum, Golgi body, lysosome, and all the functions of all these cell organelles we have learned. And the remaining cell organelles we'll learn in the next class. Thank you.